The theory of the DNA RNA code of life section. Now, as always, we need to practice. That section of the work is basically study work, but you won't be able to answer the questions properly if you don't practice. It's more or less like being a coach for a netball or a rugby team, and if you are a player, your coach will always say, we have a game plan. This is what we need to practice. When this happens, we throw the ball this way, or we have this plan that we have. So it is the same with life sciences. It doesn't help you just to study the work. You need to practice the work as well, so that you can understand what is the game plan, so that you can understand how they will ask you in the final exams. So they must say practice makes perfect. So let's practice a few short questions and section B questions. I'll try and give you some tips on how to approach some of the questions, so hopefully it helps. We were lucky enough last year. I'm going to just show you how the book looks. We received this from our ALEFs. Question banks, and what makes this bundle of joy very nice to use is that all of the questions were grouped according to the topic. So you can just look for the short questions and then the section B questions, and the learners can practice this in the class. If you don't have books like this, I'm sure you can ask other teachers, your neighboring teachers or schools, or you can just contact your learning facilitator. I'm sure they will assist you with this if you don't have it by now. So let's have a look. Let's look at the first questions, the short questions. Normally learners don't do too bad in this, but let's just have a look. Let's look at the approach. Okay, I don't have to tell you anything about how this works. You have a question and then you have to choose between certain options, A, B, C, and D. Now, normally what I tell my learners as well, when you look at the question, like this one, which of the following shows the process of DNA replication in the correct sequence? Now, I like to work with color, so here we go. So this is about DNA replication, excuse me, and the correct sequence. Now, immediately I know what should this be starting with. DNA double helix unwinds. So there I can see in A, DNA double helix, okay, so that is a possibility. 2 DNA double helixes, no, it's not, that's the end. We have 2 DNA strands at the end, so it cannot be B. So if you are sure about a certain answer that can be excluded, you exclude it immediately so that you do not confuse yourself later. And then C, I see strands separate, totally won't be one of the answers. And then I see, okay, number D, also a possibility. But now I have to go further. I have to look at the correct sequence. And when you read further, you're going to see that this one is not correct if you have studied your DNA replication sequencing. So this one is not correct, so our answer will be number A. And that is how you can approach each question. There's enough time to do this. All right. Now the next question is regarding this specific picture. They tell you question 1.1.2 and 1.1.3. They're going to ask you questions regarding this. Now, whenever you have a picture or a sketch or whatever, look at this first before you even look at the question. So I'm going to have a look. Here they say this is now a crime scene. And these are DNA from the suspects. So we must now discuss this. This is DNA profiling. I can see the bars that they talk about arranged in a specific way, the black bars. So that is DNA profiling. I see here at the crime scene there's a DNA, and there I have suspects. So immediately I can, if I want to, have a look which ones are the same. And it's like when you were in primary school and your teacher asked you to spot the difference. So here you can see that is exactly the same. So let's see what they ask us. The first question is, the procedure shown above is called what? Now, okay, that is an easy question. You just need to know that that is DNA profiling. So the answer is number C. Now, 1.1.3, if you can remember, it's still about DNA profiling. But now this is a different type of question where they list things and then they ask you to give the correct combination. So before I look at this, I don't look at that. I look at the question first. So I see, they say below is a list of possible uses for the procedure. Now we know the procedure now is DNA profiling. And now we look at the different options and we identify them. I work the same way with this that I normally do with multiple choice. So those ones that I think is correct, I mark them for myself, and those that I think isn't, I scratch them out. Otherwise, you just confuse yourself anyway, because most of the learners, you are most stressed when you write exams, and then you must forget things easily. So let's look at this. DNA profiling, do we use it for paternity testing? Now, if you can remember, paternity means father, so we're testing to see who's the father. Do we use this for DNA profiling? Yes, that is relevant for DNA profiling. Then the second one, matching of tissues for organ transplants. Now, this is a little bit wider than what we discussed, but if you use your brain, you will know that tissue will have DNA, and normally when people need to go for organ transplants, they look at their family to see who will be able to donate the organ for them, because with organ transplants what happens is that your tissue can reject the foreign tissue. So we want tissue that is more or less almost the same. And obviously then we have to do a DNA profile to see if you are compatible. So yes, that will also be an answer. This one, identification from your fingerprints. Where will you find your fingerprints? Your fingerprints are on your fingers. It is not inside cells, it's on the outside. So identification from fingerprints, it has nothing to do with DNA profiling. So you can scratch that one out already. Okay. Then lastly, screening for genetic disorders. Yes, because we want to see if there's something wrong, and then if possible we want to find cures. So now I see I have 1, I have 2, and I have 4. And now I just look for the possibilities and I see, aha, it is this one, that is the same. So the answer will be number B. Okay, 1.1.4. We have a sequence, and immediately when I see that sequence of nitrogenous bases, you still remember, we have, I'm just going to do it quickly, we have a phosphate there, we have a sugar there, and there we have our nitrogenous base, and that is the building blocks of DNA and RNA. So here in this question, they don't tell us if it's DNA or RNA, but there's a clue. Have a look, what do we have here? We have a U, and a U is for uracil, and uracil we find where? We find that in RNA. So immediately I have a clue without them telling me directly. Now I have to look. Possibilities. The first one, a gene that codes for one polypeptide. Now the moment when I see polypeptide, I think about amino acids. Because when I have amino acids combining to form a protein, what do I have in between them? I have peptide bonds. So the moment they talk about polypeptide, peptides, then they are talking about amino acids. And when we represent amino acids, we don't use coding like that. The coding we use for RNA, either tRNA or mRNA. So we will have words like this, for instance, that represents amino acids. So when they talk about polypeptides, I know, this can't be relevant for this one. So A, no. Then B starts with a DNA. So immediately I know this is not DNA because there is uracil, so this one can't be correct. C, they talk about a polypeptide again. And peptides, we just discussed. Polypeptide contains amino acids. Those are not amino acids, they code for amino acids. Okay, so D, the last one, this is an mRNA strand that codes for 5 amino acids. Yes, because that is 1 mRNA, 2, 3, 4, 5 mRNA codons that forms 5 amino acids that will form a protein at the end of the day. So the answer here is number D. Okay, then 1.1.5 is an easy one. They say the DNA of different species only differs in what? A, the components of nucleotides. No, the components of nucleotides will not differ. They are always, here we have it, the building block, the monomer. They will always be phosphate, sugar, nitrogenous base. So the components are not going to be different. The sequence of the nucleotides, the T and the A and the G and the C, the way that they are forming, the sequencing gives us different species. So this answer will be B. All right, now here we get to a nice question. Difficult for some, but I'm going to show you now how to make it easier. They say, one strand of DNA molecule. Okay, so now I have one strand. Remember, normally we have a double strand for DNA, but here they just now talk about one strand. That is why you also need to practice to read. You shouldn't just see the word and not listen and understand what the word means, because it gives you the clue. One strand of DNA contains 60 adenine. So I write it here for myself. In this one, there is now 60 adenines on this backbone of the one strand of the DNA and 20 thiamines. So I will have, when I count them, I will have 20 adenines, I'm lying, 60 adenines and 20 thiamines. Now they're going to ask you how many adenine molecules are present in the double-stranded DNA. Now they want to know how many is in the whole thing. And that is where you need to know that adenine will always combine with thiamine on that side, and thiamine will always combine with adenine. So if I have 60 adenines in this strand, I counted them, they gave it to me, and adenine combines with thiamine, so I'm going to have 60 thiamines now, but that's not relevant. They want to know about the adenines. So if I have 20 thiamines here, 20 thiamines is going to combine with 20 adenines on this strand. So now I can say 60 adenines from the first strand, combining the 20 adenines, counting them together, it's going to give me in total in this whole molecule 80 adenines. So that is why the answer will be C. So just remember to read. Don't immediately get a fright and say, what is this mathematics now in life sciences? They will not ask you something if you can't find the answer somewhere, either in your brain or on the page. So that is the multiple choice. Remember, multiple choice always counts 2 marks each in life sciences. Okay, so this is now a part of section A. The next section, the terminology, many learners find this difficult because of language. So we need to practice this quite a lot. They need to read and they need to write. So the first one, they say the organelle in the cytoplasm, which is the site of protein synthesis. Now, protein synthesis, there's 2. We have transcription in the nucleus, and then we have translation on the ribosome. So the organelle that they are talking about is the ribosome that they want. All right. The next one, the name of the bond. Now immediately when I think about DNA and protein synthesis, I think about 2 types of bonds. Now actually 3. We have phosphate sugar bond, we have the weak hydrogen bond, and then we have the peptide bond. Now they're talking about the bond that forms between the amino acids. So the one that forms between the amino acids, that one is called a peptide bond. And please remember, when they're asking the question, or they say the name of the bond, then it's not always necessary to write peptide bond, because bond is in the question already, so you can just write peptide. All right. 1.2.3, the process by which DNA molecule makes identical copies. We need that for mitosis, for instance, as well. That is identical copy is making a replica, so replication, DNA replication. That's the name of the process. This is study work. 1.2.4, the type of nucleic acid that carries a specific amino acid. Now, ladies and gents, nucleic acid, there's most 2 types of nucleic acids. we have RNA, and we have DNA. DNA cannot leave the nucleus. It can't carry anything. So what is carrying the specific amino acid? That one, there's the protein, and it looks like a clover shape with the 3 nitrogenous bases at the bottom. There's the amino acid, there's the molecule, there's the anticodon. So what do we call this nucleic acid? It is the tRNA. Okay. Type of nucleic acid that carries a specific amino acid is tRNA. Okay. 1.2.5, the nitrogenous base found only in RNA molecules, that will be uracil. Now, many times the learners say, yo, ma'am, I don't know how to spell that. Now, there's another tip. It's very easy. How do you spell uracil? Let me just show you. You are a silly child not to know how to spell uracil. So you can make a rhyme for yourself. You are a silly child. This is how you spell uracil. Don't worry about that. You can spell uracil. Okay. Now, 1.2.6, a sugar molecule found in a nucleotide of DNA. DNA, there's only one. That's why it's called DNA. It refers also to the sugar. Deoxyribose is the sugar that we find in DNA. And then lastly, the triplets, 3 babies, 3 nitrogenous bases. What do we call the triplet on the tRNA? That is called the anticodon. And please, ladies and gents, when you write tests or exams, the teacher will be very grateful. You just need to try. Don't leave open spaces. Sometimes there's a idea in your mind, and then you think, no, it can't be right, and then you don't write it. Write it. Rather take the chance than writing nothing, because if there's something, we can still look for marks. If there's nothing to mark. You have nothing to lose. Write an answer. Guess if you don't know. At least maybe you will be correct, because we tend to study and then we do know the answer, but we just confuse ourselves sometimes. All right. Now 1.3. The last type of questions that we will get in the short questions for life sciences, that is now fit column A to column B. And here in the instructions, it's even printed in bold. And many of us, we just ignore that and start answering the questions. So have a look. They say when you answer, that is the instruction that they give you. Answer A only, B only, both A and B, or none. You don't use anything else, because the moment you start improvising, you confuse the answer, and then maybe you lose marks for something that is quite easy in the final exam. Okay, so now let's look at the questions. The first one, they say pairing of nitrogenous bases. Nitrogenous bases will be adenine, guanine, thymine, and cytosine in DNA. Nitrogenous bases in RNA is adenine, uracil, guanine, and cytosine. So we have pairing of nitrogenous bases for both of them. So the answer will be both A and B. And that gives you 2 marks. The next one, 1.3.2, they say produce the first X-ray pictures of DNA. Now, if you remember, there's only 2 names that you need to put in your brain when it comes to DNA, and that is Watson and Crick. Now, these two guys here is Darwin and Lamarck. They add this because when you write your final exam, that's the 2 other names that will stick in your brain. That is regarding evolution. And Lamarck most said, the more you use something, the better it becomes. And the less you use something, the less or smaller it becomes. So use your brain here, then you will remember Watson and Crick is the answer. So this one is actually none. All right, then 1.3.3, biotechnology, processes where we use biology in technology, and that is genetic modification and cloning. So it's both A and B. So this is approach for the shorter questions. Total in this instance is 25. Hopefully you are doing well. Okay, now we're going to look at section B. Section B is now the longer type of questions and the scenarios. Okay. Now the first one we have here, you obviously will recognize that arrangement of black bars that we call a DNA profile. So please read all the information. Don't skip or don't think you just glance quickly. Many times there's information that you need. So this one, it says Mrs. Gill had baked a birthday cake for her sister Anna-Marie and kept it in a tin on the kitchen table, which all of us know is actually very stupid, but anyway. When she returned from shopping, all that was left in the cake tin was a few crumbs and then a smudge of blood where the thief had snagged his or her finger on the sharp edge of the tin. Now they say, study the DNA profile of her 4 children given below. This is quite a mother that takes a DNA profile of children that's stealing cake, but nevertheless, let's have a look. Here is the first one, the blood sample. This is now our proof, the DNA profile of the blood sample. And then we have Bob, Sue, John, and Lisa, the 4 children. And now we play the game that we did in the primary school again. We see which ones match. And I can see, when I look here, that we have a culprit. There is our blood sample, and there is the culprit. And the culprit, or the hungry child, seems to be John. We see that that is similar. So now we look at the questions. Let's have a look what they want to know. They say 3.1.1, define the scientific term for DNA profile. Now immediately the learners in my class, when they see scientific, I don't know, what is this? This is not science. It's not physical science. What are they asking me? Remember, life sciences are also a science. So if the word scientific confuses you, you just do this and you say, define the term DNA profile. That must now sound more familiar. And the moment they say define, they must now want a definition. And let's look at the mark allocation. It's 2 marks. So now you think, what are you going to write? You can't just wobble, you must write the fact. So what is a DNA profile? We can say it is an arrangement or a pattern, arrangement of what? It is an arrangement of black bars. So actually you have the answer here in front of you. You just need to use your common sense. Arrangement of black bars of what? Of a DNA segment. Okay? And what do we use it for? We use it to compare it with a known profile. So that is what you write. There we have what it is and what it is doing. It gives you the definition of a DNA profile. Okay. Now 3.1.2, they say which one, and when it's written like that, they're just going to mark the first answer. They're not going to look for the right answer. They mark the first answer. So remember, whenever in a question paper they have the words in capital, 1 or 2 or 3, it means that the teacher will only mark the first 3 answers. So don't think your teacher is going to look for the answer. Think about what you want to say and then write it. Also, don't summarize it. Then we summarize your marks. You have to write a full sentence. Okay, so which one of Mrs. Gill's children is most probably the suspect? Now that we have, we know it's John. So that is your first mark, John. Then they say, give a reason for your answer. How do we know it's John? We know because the DNA profile of John matches the DNA profile of the blood sample. Now you can't be lazy. Many learners just say, it matches. What is matching? Your outfit with your girlfriend's outfit or what? What is matching? And you have it there. The blood sample, the DNA profile of the blood sample matches, that's the word, or it's the same, or it's exactly the same as the DNA profile of John. Okay, so if you want the marks, you have to do the work. You have to write. Don't think, oh, but the teacher knows what I'm talking about. You must explain it as if the teacher doesn't know a thing. All right, now 3.1.3, here we go. Describe how DNA profiling can be used for paternity. What is the word paternity again? Pa, dad, parent. Okay, so paternity test is to see who is the dad, who is the dad. So how can I use this in DNA profiling? So it's for 2 marks. So we can say that the child's DNA profile is going to be a combination. It's going to be a combination of what? Combination of both parents. And because of that, we can use that as proof. So what are we talking about? We're talking about the child and the child's profile. We're busy with DNA profile. And how can we use it for paternity testing? Because the mother and the father's DNA profile will be a combination of the child. That is why. Child DNA profile, combination of both parents. There's your 2 marks. Okay. Now a little bit of application in 3.1.4. Do you think that saliva, spit, saliva, can be used as a sample material in this type of investigation? Motivate your answer. So if you don't know, you can guess. You're 50-50, yes or no. That's your possible answers. Well, hopefully you guess yes, because that is correct. Saliva can be used. And why? Because we can find DNA in saliva. So saliva can be used for DNA profiling because we can find DNA in saliva and we can make a DNA profile. Okay, so that is then question 3. Now we're going to questions that's a little bit more difficult regarding that big process of protein synthesis, the making of proteins. Now, let's have a look. They say the table below shows the DNA-based triplets. Now that is why you need to know terminology. Where am I going to find the DNA-based triplets? Let's just get our GPS working again. Here I have my nucleus. There I have the ribosome in the cytoplasm. All right. Inside the nucleus, we are writing the message. So this is transcription that is taking place here. Outside, we need translation of this message. We need to understand what is the protein that we need to make here. So this process is translation. We have the DNA strand here inside this one, the DNA strand, and then we call this DNA strand that we use as the template. We call that the DNA template, because we're using that as the recipe. And then the first 3 nitrogenous bases, we call the DNA base, because it's bases, triplet, because there's 3 babies, triplets. So the first 3 of them is the DNA-based triplets. Now let's look at the question again. They say the DNA-based triplets are listed here. So they give us 1, 2, 3, 4 different base triplets. So they will be 1 triplet, 2 triplets, 3 triplets, 4 triplets. So in your mind's eye, you know now where you are. All right. Now they give you the DNA templates, the base triplets, and then they have something else here, monomer X. And then they have names. Now these names are the names of amino acids. You don't have to study the different types of amino acids. The moment they ask you about amino acids, they will give you the names. They will provide you with the names. So now there's a reason why they have this word here, monomer X. They want to check if you know what the word monomer means. Now monomer is nothing else than a building block. Okay? So the building blocks of DNA is nucleotides, but I don't have a list of nucleotides. I have a list of amino acids. Now, amino acids are the building blocks of proteins. And now they ask you, what is this monomer, this list of monomers? X is most, you don't know what it is. So they want you to know, what is this now? So this is a list of amino acids. And now we look at the questions. 1.1.1, where would the DNA template be found in the cell? That we must know now, because DNA can't go anywhere, it stays in the nucleus. So the answer, where will we find the DNA template? Obviously, That will be in the nucleus. So they always start with the easy questions and then it becomes more difficult. Then name so this whole section is now about monomers. Name the monomer X. Now at first glance you're all going to think it's very difficult, but I just explained to you that's the amino acids. So the answer here is amino acids. Okay. Now they want to know, what do we call the bond that forms between these monomers? And what do we call the bond that forms between the amino acids that's forming a protein? We call that a peptide bond. So you need to be wide awake here, because if you don't know what is the monomer, you are going to be struggling to answer both these questions, and then already you lose 2 marks. This is just terminology, this is just reading and not panicking, remembering that you do know what is happening here, you can find the answer. Okay, now it becomes tricky. They say they want the codon for tyrosine. Now where in the world is tyrosine? Here I see the word tyrosine, and this DNA template codes for tyrosine. So they are now trying to trick you to see if you know your terminology. So the codon is the term for the 3 nitrogenous bases that we find on the mRNA. Okay? Tyrosine is the amino acid that is formed. So what we can do here is we can just write it out. For instance, we start with DNA in the beginning. We have DNA. Then the mRNA comes, the codon, and it codes according to the DNA template. The mRNA goes to the ribosome, it lies there. Then the tRNA comes with the amino acid to code according the codes, the anticodon and the codon combine there, and it brings the amino acid here. So what do I have here? They are asking this one, and in that template I have the DNA, I have this one, and I have that one. So now they're asking me this one. How am I going to get this one? I can get the codon if I have the DNA. And do I have the DNA? Yes, I have it. It's here in the table. They gave it to me. So I just check tyrosine, the DNA template, the start for tyrosine here is ATA. But now remember, I'm working with DNA, so there's going to be a T. But this one is mRNA. So there's not going to be T's. There will be uracils. So let's have a look. The adenine is going to combine in the codon with the U. And then the next one, the T, is going to combine with A, and that one is going to combine with U. So the codon for tyrosine will be UAU. All right, next one, again, now they're checking your terminology. Anticodon for cysteine. Where do I find cysteine? There. There I have the template, I have the DNA. Now they want the anticodon. The anticodon is on the tRNA, okay? So now, again, we start again. Let's do it on this side. Now the amino acid is, what I have here is cysteine. So cysteine starts with the coding ACA. And now they want that one. So first I have to get the mRNA. It's more a process. So the mRNA will be UGU. And then when I find the tRNA, it will be ACA. So the answer is ACA. And we're lucky here, it only counts 1 mark. For a lot of work, it's only 1 mark. But if it counts, for instance, 2 marks, remember, you can't get 1 for effort. You have to have the correct whole sequence. To get UAU, the mark, you must have all of it. So either you will get 2 or you will get 0. There's no in-between mark. So practice these things. As sure as I'm sitting here, they will ask you things like this. It will be in the exams because it's challenging and it makes you think, and we have to distinguish between easier and more difficult questions. All right. Still busy with protein synthesis. What did I say when you have a picture like this? Don't worry, we're almost finished. This is the last question. you have to get your GPS working. So that is the nucleus. This is now inside the nucleus. So the process that's happening here, according to me, when it's inside the nucleus, is transcription. I'm not even looking at the questions yet. I'm just getting my GPS working so that I know where am I in the question, what is happening. So this one is the ribosome. There's my ribosome for the protein synthesis. And then process B, obviously this process that's taking place here will be translation. Okay? And then I see some more letters. Obviously they will maybe ask me, what is X then? That will be an amino acid. That will be an amino acid. And that is an amino acid. That's tRNA, tRNA, tRNA. This will be an anticodon. So here I have my mRNA coming from the nucleus. Here's the arrow showing it's coming from there using the DNA template. And then 3 of them will be the codon. So I'm just telling myself again what is happening here. Okay, let's have a look. They're talking about insulin. Insulin is a protein. Insulin is needed to keep your blood glucose level constant. And they are now saying that this process is making insulin. Okay, now I know what's happening in the picture. Now I look at the questions. All right, number 1. 1.2.1, name the following. What is molecule X? This is now a molecule. There I have X. Let's just check again. And there's the bracket. So it's talking about that thing, that molecule. And what do we call that molecule? That is tRNA. It's bringing the amino acid, so it's tRNA. Okay? Then the next one, process. So there I have a process and there I have a process. Okay? So process B will be translation. And now I don't confuse myself by stressing and wondering what is happening. I must already sorted myself out there at the top. Okay? All right, now they say in 1.2.2, describe process A. Now process A is this one. It's the one inside the nucleus. And the moment where they say process A, you can get a mark immediately if you say what is the process. So what is our process? The process, you can say process A is transcription. And then you discuss transcription as you have studied this directly from out of the guidelines, word for word, that whole section where the DNA double helix unwinds, okay? The weak hydrogen bonds break and then the strands unzip. I'm not going to write everything out, you need to study this. It unzips. And then the free nucleotide bases from the nucleus come and combine. A is going to combine with U and C with G because I'm working with mRNA, if you can remember. And then you will say the mRNA strand will now move towards the ribosome. It's moving out of the nucleus, it's going to the ribosome. But this is 100% in your guidelines. You study that exactly like it is from the guidelines. We are lucky. Sometimes they ask transcription, sometimes they ask translation, and that is basic study work. All right. Now, again, 1.2.3 can be a little bit tricky because, again, it's sort of mathematics. They say a sample of DNA was analyzed and 28% of the nucleotides contain thiamine. So I'm just going to work here so that you can see. The DNA that we're talking about, there's 4 nitrogenous bases. We have thiamine, we have adenine, we have cytosine, and we have guanine. And the moment they're talking about percentages, you must know percentages will be out of 100. Okay? So how many thiamine do we have? We have 28% thiamine. Now, common sense tells you, because this is what you've studied, if I have 28% of thiamine, I must also have 28% of adenine. So when I count the 2 together, the 2 of them, it is 52%. So they make up 52% of the 100. 100 minus 52 gives you 48. And then I just have to divide this by 2. Okay? Let's just see if I'm counting correctly now. Yes. So half of that is 2, half of that is that. So now that one will be 24% and that one will be 24%. And let me just check 16. No, that must be 8, it's 60, yeah, 40, 48, yeah. So the same amount of cytosine and the same amount of guanine. Now they say calculate the percentage of nucleotides which contain cytosine, and then that is your answer. Okay. They will always ask questions like this because they want to make sure that you know that the amount of thiamine and the amount of adenine are the same. They combine with one another. Cytosine and guanine are the same. They combine with one another. As you can remember, we had a question like that, a little bit different, but the same kind of thing in the short questions, in the multiple choice. Okay. Now, the next one, 1.2.4. Describe how DNA mutations will affect the structure of the protein. Now, a mutation is a change in the sequence of the structure. So, for instance, let's say, for instance, now, this DNA sequence, it codes for this mRNA, but now instead of having UAC here, there's a nucleotide that comes in, a mutation, and it just fits in there, and we have AAC. So the moment that happens, this is going to change the codon of the mRNA, which is going to change the anticodon. The anticodon that must combine there will now not be CAU, it will be UUG, for instance, if that now changed. And the moment it changes the tRNA, or the anticodon, it's also going to change the amino acid, and sometimes when it changes the sequence of the amino acid, it will change the protein. And that is why it's 4 marks, because you have to describe the process. What is a mutation, what happens, what will change, and what will be the snowball effect at the end of the day. All right, 1.2.5, tabulate 2 differences between DNA and RNA. That's also not difficult. So first of all, remember, when they say tabulate, you get a mark for the table already. Then it counts 5 marks. It means you have to give 2 differences between DNA and RNA. And that is, for instance, DNA, the sugar that we use is deoxyribose. The sugar in RNA is ribose. DNA is a double-stranded molecule. RNA is a single-stranded molecule. There's a long list. You can use any of them. So it's quite easy marks there. All right. Now, we're getting near to the end. Again, we have a diagram regarding protein synthesis. Very important, ladies and gents. There's the nucleus. Where are we? What are we doing? That is the ribosome. Okay. There I can see there's my DNA. This will be the mRNA. That will be the codon. And then it comes out of the nucleus. This is transcription. And this one is translation happening outside. And here I see that is our amino acid. That is our tRNA. This is an anticodon. So I just make notes for myself so that I know what is happening here. First question, the molecule represented by N. N is that whole thing. What do we call N? mRNA. Okay, so that's the messenger RNA. That is the answer there. 2.1.2, write down the sequence of the first three nitrogenous bases on the DNA strand, led by the formation of Z. Now there is Z, so this is what they give me. That one is the mRNA, messenger RNA, and that is UCA. It is given to me. Now they want what is there, what started this message. They want the DNA, so they want the DNA triplets, the base triplets. And remember, I'm working with DNA. The moment I'm working with DNA, I don't have uracil anymore, I have thiamine. So that will have combined with an A, C with G, and that would have been a T. So that is the answer for 2.2. And here you have your 2 marks. You see the mark allocation. So either you get 2 marks or you get zippity doodah 0. You have to have this correct. You see it's again the same kinds of questions. So you will get this. You have to practice this. You have to practice your game plan. Remember. Okay. Now, in this next section, I'll try to balance it for you because the questions that's asked here, you have to look at this picture as well. So they say, the table below, here we have a table again, shows the base triplets. You remember the base triplets? They are the 3 babies at the DNA, Moose, remember? Here they are, the base triplets, that's what is starting everything. And there I have the amino acid, there I have what is happening at the end. So if I look at this picture, in the table, they are giving me the base triplets that are here, the 3 nitrogenous bases adjacent to one another that we start with, and then they give me the amino acids, so they give me what we end with here. So I'm sure they're going to ask us about what is happening in between. They're going to ask about the mRNA and the tRNA. So now you can also tell us what's going to happen in the future, because that is what we are guessing what is going to happen. What will the questions be? So let's have a look. 2.1.3, with reference to the diagram in question 2.1, this is now the diagram, remember, and the table above. This is now your resources that you're going to use to answer the questions. State the base sequence of the molecule labeled Q. Labeled Q. So let's look for Q. Yes, Q. So Q is going to be a tRNA molecule. Now they want to know what is the base sequence here. How am I going to know what is that? I don't have, for instance, amino acid there so that I can use the DNA. But what do I have here? Here's my clue. Here I have GGA, and that is the mRNA. So the mRNA is our clue. The mRNA is GGC. So what will be the anticodon that's combining with GGC? Remember, we're busy with RNA, so G is going to be C, G is going to be C, and combine with that one. So our answer is CCG. All right. Next one. Name the amino acid labeled P. Okay, there we have the amino acid. It is on this DNA molecule that you see here. And the only clue I have here is the sequence of the anticodon. So now I write the story for myself again. They want the amino acid, so they want that one. Before I get the amino acid, I have a tRNA that they give to me there. They say UGU. Now I have to find the mRNA. When I have the mRNA, I can find the DNA. And when I have the DNA, I can identify the amino acid. So I just follow the sequence. Okay, tRNA, UGU, I got it from there. There it is. Now, what is the mRNA coding for that one? Obviously, that will be A combines with that one, C and A. That is the mRNA, the message that's going to be made according to the DNA. The DNA will be T, G, T. So now I have the DNA. Now I can use it. I get it from there. Do you see a T, G, T there somewhere? Let me just move it. Yes, there's a T, G, T. And what is the name of the amino acid? 309. 309. 309. All right, that's the answer. So that is the process. Just stick to the recipe the whole time. Stick to the sequence. Find your procedure. Don't take shortcuts, otherwise you make mistakes. Okay. 2.1.4, describe how the composition of the protein molecule changes if the base sequence at X is UGU instead of UCA. Okay, now let's have a look here again. They say, here's the base sequence. This is mRNA, remember. The base sequence is UCA. That is what it begins with. Now, what is going to happen to this whole process if that UCA, because of mutation, changes to UGU? Now, let's have a look. Let's have a look what happens. I'm still going to use that sequence. All right. So the mRNA that I have there originally in the question, UCA. So now the UCA, what is the DNA that it's going to combine with? It will be AG, it's DNA, so it's T. Do I find, let's just go there, do I find AGT? Yes, there's AGT. So that original coding of UCA will give us the amino acid of serine. So that's the amino acid that we will get. But now they say it's not UCA, it's going to change to UGU. So now let's test what happens. If the mRNA is now UGU, what will happen to the DNA? I will have ACA. Do I have an example like that there? Yes, there's ACA. So what is the amino acid now? Now it's cysteine. Now when you answer this question, they're asking you how the composition of the protein molecule changes if the base sequence changes. And then you say the composition of the protein molecule changes because the amino acid serine is now not the amino acid that is forming anymore, it's now cysteine. So you get your 1 mark there and 1 mark there for explaining what is happening. All right. Last question. Name and describe the phase of protein synthesis taking place at the ribosome. Which one is at the ribosome? That is the translation. Again, they say name the phase. So when you say this process is translation, already you have a mark. Okay, so according to your guidelines that you have received from your teacher, you study translation, transcription to the letter in the guidelines, and then that is just what you produce now. You will say the mRNA molecule attaches itself to the ribosome. The tRNA molecule, the anticodon of the tRNA molecule comes to combine with the codon of the mRNA molecule, and the tRNA molecule brings a specific amino acid, and according to the mRNA sequence, all those different amino acids combine next to one another. They form a peptide bond in between, and then it forms a protein molecule which is going to curl up, be a protein, and have a function. So ladies and gents, I hope it helped you a little bit. Just remember what I said. You need to have a game plan. You need to practice. You cannot just study the theory and then think you will be okay. You have to practice papers. I know some of you find it difficult, but you have to put your buttocks on the chair, sit still, try this, and if you don't understand, please ask your teacher to assist. I'm sure your teacher will be more than willing. Good luck and practice a lot. You will be happy.